Okay, so I'm going to present our last study on the impact of genetic heterogeneity and pleiotropy in psychiatry on brain functional connectivity. So, as you might know, psychiatric condition has been mostly studied through a top-down approach, starting from the psychiatric diagnosis and moving down to clinical cognitive dimension, brain endophenotype, and genetic risk, like to identify the gene of autism, like the gene of schizophrenia. But so far, research using the top-down approach identified large clinical heterogeneity, a lot of comorbidities. At the imaging levels, they were not able to identify clear biomarker. They reported small effect size and a lot of heterogeneity and overlap of signature. And finally, it can make sense, this imaging findings, if you look at the result at the genetic level, because so far, more than 1,000 mutations have been associated with autism and more largely like with neurodevelopmental condition. And on top of that, 6,000 additional genes are proposed are candidate gene for NDD. All these genes are located all over the genome, as you can see on the right side. And in addition, we have a phenomenon called pleiotropy, where the same gene can be involved in multiple diagnoses. So basically, in one subject, the gene A will confer risk for ASD and for schizophrenia in another subject. Okay. So this pleiotropy has been quantified at the common variant level using GWAS result, a summary of GWAS. So for example, in this study from the Psychiatric Genomic Consortium, they correlated result from GWAS of ASD, ADHD, etc., 8 psychiatric conditions, and they found interrelated disorder at the common variant level. In the same way, in the same manner, the researcher has attempted to quantify the relationship across disorder at the gene expression level, so the transcriptomic correlation, transcriptome correlation, and they correlated this result at the gene expression level with previously observed result at the genetic SNP-based level, and they found a good concordance between genetic and transcriptomic scale. And we wanted to know if these observations at the genetic and transcriptomic scale could be similar at the connectomic, functional connectomic scale of observation. So for this purpose, we use 4 psychiatric condition datasets, so publicly available. So ABIDE 1 and 2, ADHD 200, COBRE, CNP, Consortium for Neurophenomics condition, and unselected population, so UKI bank, to identify the effect of 2 traits, fluid intelligence and neuroticism, and the control condition, which is the inflammatory bowel disease. So we performed like classical standard preprocessing with NIAC. We did parcellation into 12 network and 64 regions. And we just then compared compare cases and control using standard GLM with age, sex, scanning site, motion, and global signal as covariate. So using this method, we were able to define signature of bipolar, schizophrenia, etc., all of our condition, and we correlated them, and then we did some permutation to test the robustness of this correlation. So we saw significant correlation between schizophrenia and bipolar condition, for example, schizophrenia and neuroticism, etc. So we wanted to know if this correlation at the connectomic level could be similar with previously established correlation. And it was the case for if we compared our connectivity correlation and the one established at the gene expression level, but we only have a few dots. But it was also the same, like with 20 dots now, with correlation established at the genetic level, so with GWAS, common variant. So for example, the correlation observed between schizophrenia and bipolar at the connectomic level was similar to the one observed at the genetic level. So we then wanted to know if this relationship between condition at the common variant level, so these R equals 0.3, the GWAS level, could be observed also with rare variant. So for example, 16p versus 22q, do they present similarities at the brain level? They are both CNVs enriched in psychiatric condition, highly enriched. And so in our previous paper, we showed similarities in this area, in particular thalamic, auditory, and motor area between two CNVs. So we wanted to extend that. So for this purpose, we added like these CNVs associated with different degree with psychiatric condition, so rare variant, and we also added common variant using polygenic risk score for psychiatric condition and control PRS. We first quantified their impact on connectivity and compared it. So we showed a medium to large effect of psychiatric CNVs, right? A really tiny effect, almost no effect of polygenic risk score. And we attempt to correlate their effect on connectivity of genetic risk with the number of point of like IQ point lost associated, like reported in the clinic. And we found a correlation, a good correlation between them. But we didn't find any effect of, any strong effect of PRS. It was still significant, but really tiny. Okay, so then we wanted to know if all these genetic risks, so rare and common, could converge from genome to transcriptome to connectome at some psychiatric hubs that could explain why you have like 1,000 of genes associated with ASD or with NDD, and or several thousand then, and only a few diagnoses. So we characterized their signature. So on the left side of our main genetic risk and PRS, and then we wanted to know if they could converge. So first you can, so the red represent overconnectivity and blue underconnectivity. Sorry. So first you can observe like the fact that every network is affected by at least one genetic risk, but still that we have a recurrent pattern of overconnectivity in basal ganglia, thalamus, and in motor networks. And this pattern was also reported in several psychiatric conditions, including ASD and schizophrenia. So we wanted to quantify that a bit better because it's only description here. So we correlated our profile between genetic risk and psychiatric condition. So for example, here you have the fixed 54 between schizophrenia and bipolar. And we wanted to know which connection were responsible of this relationship. So we did a PCA and we showed that the connections that were mostly involved in this relationship were the overconnectivity between, again, the basal ganglia thalamus here, with somatomotor area and auditory area. So the first dimension explained like a quarter of the variance. So basically, to illustrate that, we I showed you here, I'm showing you the profile of the thalamus. So if you take the profile of the thalamus, in several conditions, you can see a recurrent overconnectivity of the thalamus with the motor, auditory, and visual area in ASD, in schizophrenia, also in 22q without the visual, del16p, but you can also observe that with a lower effect size, but still in PRS associated with MDD, in neuroticism, and not at all in G factor, as you can see here. So to conclude, we showed that pleiotropy and polygeny drastically impact our ability to detect clear biomarker signature of psychiatric manifestation. We also saw that despite this heterogeneity, we were able to identify a critical and recurrent contribution of the thalamus and of the somatomotor network across the genetic risk and condition, psychiatric condition. So we suggest that future study need to both do a partitioning of polygenic risk score, as well as to cluster rare variant based on gene function. So for example, their application at the synaptic level to obtain mechanistically coherent subgroup of individuals. So I want to thank my lab and my colleague from my new lab. Thanks for your attention.